and detect that cancer.
Hey everybody, it's time for This Week in Science and Education. This is your host Kevin Kugler from the Virtual Researcher on Call program. Thanks a lot for joining us on another episode of This Week in Science and Education. First of all, let's welcome my regular co-host, Colin Jago from the Kawartha Pine Ridge District School Board, and Dr. Thomas Merritt up there at Laurentian University. How's it going, fellas?
Very well.
Yep, very well.
Good stuff. Beautiful day out, and we've got a fantastic show today. We're going to learn a little bit about some cancer research, and speaking of that, let's introduce our guest. This is Dr. Michael Kolios from Ryerson University. Dr. Kolios, thank you very much for taking the time to join us today. We appreciate you taking some time out of your day. I know it's very busy as a cancer researcher, but on behalf of the kids, they all appreciate it. So thank you, and welcome to the show. Let's get it started by just having you give us a quick little, minute, 2-minute catch up on exactly what it is that you do.
Okay. So thank you first for inviting me to talk, right? It's a great opportunity to let people know exactly, what happens in people's labs. So what I do in terms of cancer research is not typical in a sense. So I use ultrasound methodologies to do a couple of things, like the same ultrasound that you would use, when imaging babies or something like that, to do two things: to try to see how cancers respond to treatment, right? And in other ways to actually treat the cancer itself. So basically, very long time ago, when I was a graduate student, together with Dr. Gregory Charnada, who's a radiation oncologist at Sunnybrook Hospital, we discovered that when cells die, and they die in a particular way called apoptosis, so that's called cell suicide, right? They scatter ultrasound in a very specific way, right? So after our discovery, we tried to see whether we could use this methodology to see if we can look at how tumors respond to treatment during an actual treatment. The big idea here being is that when somebody's given a cancer treatment, usually that is over weeks, if not months, and typically the radiation oncologist, doesn't know whether during that time period the patient is responding to the treatment or not. So what we want to do and what we've developed is an ultrasound technology to look at how cells scatter the sound, in this case cancer cells, but especially when they start dying due to the chemotherapy, right? If we could detect that non-invasively with ultrasound imaging. So in our experiments, typically what we do is we take these kind of cancer cells, right? We just shoot ultrasound on them and see how the waves scattered back, what they look like, right? We have these kind of mathematical tools which we analyze the signals that come back. And ultimately what we want to see is if we could use this reliably as a tool to see if patients are responding to chemotherapy just days after the chemotherapy has started, not weeks or months. So that's kind of encapsulates the basic idea.
Okay, perfect. Go ahead, Thomas.
So, Michael, from a scientific point of view, it's a really amazing tool that you can do this. How do we end up applying this? So there's a researcher here at Health Sciences North, the hospital that's associated with Laurentian University, that is developing a similar kind of system using RNA quality. And one of the questions that I've had to him when we talk about it is, how do you use this? So you've got a non-invasive way to monitor the efficacy, I guess, of treatment. How does a clinician use that information?
Right. So if you actually, we've been doing this work now for 20 years, right? So this has gone from the test tube to the small animal. And actually just a few weeks ago, we had our first clinical paper published. So these are women with locally advanced breast cancer. It's a very aggressive cancer that unfortunately a lot of young women get. And of critical importance is knowing whether the chemotherapy they're given, right, if they're responding to that or not. And so we're using this ultrasound methodology currently, right, and we've published on it, and we see that we seem to be able to detect whether patients are responding to treatment using this ultrasound methodology. So, of course, we're not altering any treatments yet, right, because we're still in the preliminary phases of showing it working in humans, but the big idea has as follows, right? A radiation oncologist starts giving a particular type of treatment, and if within a week or two, with the ultrasound technology, they don't see the ultrasound signatures of a response to chemotherapy, then they will switch.
Yeah. Okay. So is it a matter of switching or a matter of going off? I mean, I know that one of the issues with chemotherapy is all of the negative side effects that are associated with that come along with trying to battle cancer. So is it simply trying to not use a therapeutic agent if it's not working, or do we have the possibility that you could switch to a different agent?
Right, so that depends very much on the particular type of cancer and how it's diagnosed, but the bigger idea is to switch, not to stop the treatment, right? So in other words, this type of treatment, for whatever reason, is ineffective. Let's go to the list of other potential treatments and see what we could do.
Right.
Go ahead, Colin.
Yeah, thank you. That's amazing. That's fascinating that you can do that. And the question of switching, I'm thinking about the speed. Like when you use your technique to measure the cells as they undergo or after they've undergone apoptosis, what's sort of the time span between like a cell in a tumor, for example, dying and then disintegrating, I guess. Like I'm trying to think what that looks like in terms of your ultrasound and how much time you have, for example, after a cell dies until it's gone and you can't see anything. Like what's that time frame look like?
Yeah, I could tell you a lot about in vitro experiments, so things that we have very good control over, right? What happens in a patient obviously is very, very complicated, right?
Sure.
But we've done it both in what is in cells, right, and in small animals. So basically getting cancer cells, injecting them in the hind leg of mice, growing the tumors and looking at it. So if you look at a lot of these chemotherapeutic treatments, usually, right, they have an effect in terms of every cell cycle or so, right?
Sure.
So the initial cellular effects clearly occur around within 24 hours to 48 hours, depending again how aggressive the tumor is and the division rate of the cancer cells. So clearly in vitro and in vivo in small animals, we see maximal responses around 24 to 48 hours. Because as you pointed out, right, especially in vivo systems, I mean, right, the immune system comes after and kind of cleans up a little bit the mess,
Cleans it
so to
up.
speak. Right. So it's quite difficult to make conclusive scientific statements about what happens in vivo, even though we have histology, right? But clearly in vitro, we get maximal response during what is called the nuclear condensation and fragmentation phase of apoptosis, right? So when the nucleus kind of condenses and then fragments, it seems like that's when the cells scatter most of the sound. So we could detect those kind of signatures. The big picture also is, let me just finish this one thought, I guess, is that we use something called high-frequency ultrasound, right? So if you look at the wavelength of high-frequency ultrasound, it's about 30 to 40 microns. So that's, a cell is about 20 microns, so it's very comparable. In that case, the technique is very sensitive. The million-dollar question for us was, when we go down to 1 megahertz ultrasound, which is the one that used in clinics, right, 1 to 10 megahertz, will the technique be as sensitive? So the trial that was published now in Clinical Cancer Research shows that we're seeing changes, right? Now, what those changes are attributed to is a lot more difficult to state. But clearly, in very well-controlled systems, we know that it is the nuclear condensation and fragmentation.
So there's a humanist element to this as well. So science aside, everybody has a story of somebody that has cancer, right? So mine is I have a family member who has had a mastectomy, and she's undergoing chemotherapy and radiation in some capacity, and I'm not exactly sure the details. But I do know that, like we were talking about, it can be weeks, it can be months, and it's the physical drain on your body, it's the nauseous and all that kind of stuff, right? If she could know after 2 weeks or 3 weeks, yes, this is making a difference, yes, I have some hope, just even knowing that gives her the strength sometimes to get through the next treatment, right? To get to the next day. This is worth it. I'm going to keep going with this because I know it's working. That's huge.
That's a good point, Kev.
There's many angles to this, right? Not only is it, sparing the person the comorbidities or, the bad effects of the treatment. There's also, if you look at many of the new chemotherapeutic treatments, they're extremely expensive, right? $100,000, $150,000 for the whole course of treatment, right? Now, imagine that it's extremely expensive to give all that, right, and it not being effective. So the other angle I'm looking at this is if we show it's robust, right, and it's a big if still, okay, then, it could have a big impact on the healthcare system by saving resources, right,
Yeah.
that are very precious these days.
You bet. Great point. Back to the science. Thomas, I interrupted you.
No, not at all. That's a great point. And Kevin, I think we sometimes miss the human side of what the research means. Michael, I wonder if you could talk a little bit about your your transition as a scientist, or the transition of the work that you're doing. So you've talked about the idea that you've got in vitro studies, so in studies in glass, in the lab, in a very controlled system, and then you're now applying them in vivo in the living organism. And you alluded to a really interesting progression where you start off in a very controlled situation, and then you move into model organisms, I guess you were saying either mice or rats, and then you're now transitioning to humans. One of the things that we'd like to do is talk to students about how science actually progresses. So can you tell us a little bit about how the work that you're doing has progressed from in vitro, in glass, now to clinical trials?
Yeah, slowly and difficultly. So let me explain that a little bit better, right? So if you look at actually even the basic premise, when we initially start publishing the first results, there was a lot of skepticism, because if you look at some of the basic physics behind this, right, it wasn't that obvious that a cell undergoing apoptosis should have any kind of special signature, right? So, we first published the results. There was a bit of skepticism, but then, for the next 10 years, we start working on because my background actually is physics, right? So I was looking at the physics of scattering, okay? So in other words, if you have a little, object and you have a wave hitting it, right, how does it scatter that wave? So we did a lot of kind of fundamental work, and we start kind of getting a better clue as to why exactly we're observing. And actually, another interesting story is how this all started. This all started over beer during a graduate student seminar, right? Where my collaborator, Dr. Gregory Charnata, the radiation oncologist, was listening to this seminar while we were both graduate students, and said, "Can you use this technology of ultrasound to detect apoptosis?" He asked the presenter. The presenter said no, but we didn't believe the presenter, right? So we had a bet, and late at night, both of us were doing experiments on ophthalmic ultrasound, so an ultrasound that's used for the eye, with, of course, the permission of the ophthalmologist, but the technician really hated it, right, because we were basically using a machine, right, on cancer cells. So we established that you could see this. We did the very rigorous experiments, and we were very fortunate to get very good funding from the Canadian Institutes of Health Research, right, which is one of the main funding agencies that funds this kind of research. And over the years, the next step, after the first 5 years that were just in vitro, right, we went in vivo. So we did many animal studies to show that this effect is robust. But even when you do animal studies, right, people say, okay, fine, you could show that it works in a small animal system, right? That doesn't mean that that could be translatable to a human system. And with many cancer treatments, that's been shown again and again. Let's say anti-angiogenic drugs, right? Drugs that are able to stop the development of new blood vessels to tumors. I mean, they showed exquisite results, right, in the late 90s, but that really didn't translate that well when it went to humans. So we're right now at the stage where we just have our first paper published on this, right? It seems that we are detecting changes, structural changes in tissue, right, due to chemotherapy. And, we're very excited because we're ramping up now the number of people and, trying to make it a much more robust conclusion. And the way this picks up is right now we're only doing it at Sunnybrook Hospital where my collaborator is, and we're now starting to expand to other centers. So the other thing you have to show is that, it's not just your center with your expertise that could do this for it to have an impact, right? It has to be, so to speak, easy enough and translatable enough so that many different cancer centers could start using it.
I wonder if one of the reasons that you seem to be having success with transferrin is that this whole the concept is based on physics, right?
Yeah.
When we look at drug response, we're just beginning to understand the genetics behind drug response in humans, let alone model organisms, or you can look at it the other way around, model organisms, let alone humans. And there's so much difference from individual to individual to individual in the genetics behind drug response. You're working with physics, right? I mean, physics is what physicists are what we all wish we were if we were smarter and were better at math, right? And
Okay.
the physics behind the ultrasound is the same whether you're zapping a rat, a mouse, or a human. And I can imagine that would make it more transferable across those. And as a geneticist, I talk to people about the idea that genetics is the same across all of these things. And at one level it is, but at the other level, it's incredibly different. So I yet again had physics envy, that I'm guessing that's why this is transferring as well as it does.
Yeah, partially, but don't forget that, it's not that simple either. So in other words, you said from cell to animal to tumor, right? Well, if we look at the type of tumors we use, these xenografts, right? The histology, because really what this depends on is what does the histology look like? Where are the structures that scatter the sound? So the histology of xenograft tumors is very different, right, than the what we would call wild type tumors you would have. So, you have to be very careful in every step. So yes, indeed, scattering physics is scattering physics, but the model systems you use might not be appropriate for what the end goal is, which in this case is we want to see if it works in humans, right?
That's an awesome point, and I think it gets to the basic idea of really understanding your assumptions. And when we
Yep.
talk to kids about how you do science, I mean, the biggest hurdle I think that we have as scientists is acknowledging and realizing where the assumptions are. And I mean, that was one I missed completely. That you're entirely correct. That tumor morphology is different. That's an awesome point. Thank you.
Colin,
No
I
problem.
have no idea what these guys are talking about at this point. Can you bring us back to K to 12, please?
Uh-oh. You know what? I'm going to do two things. I'm going to tell you a story that I love telling this story, and it reminds me of what you just said because the point where you said we asked the presenter if you can detect scattering from cells that have undergone apoptosis, and you said no, and you didn't believe him, right? Apoptosis. There we go.
There you
And
go.
you didn't believe him. I think that's amazing because I remember 3 years ago I talked to a student at the Canada-wide Science Fair who had done some science fair research on looking at various medicines to help cure cancer, right? And she had gone to her teacher and said, "This is what I want to do," and her teacher said, "No, you can't do that. It's too hard. You can't do that. You can't do that." She then proceeded to contact, I think it was 45 or 50 different researchers, finally found someone willing to humor her, went into the lab, did some work, ended up at the Canada-wide Science Fair. So she, again, ignored someone who told her she couldn't do this because she thought that there was something to it. So it's a good story, and I think it's one that postgrads can resonate with. I also think high school kids can also resonate with it as well.
Yep.
I'm wondering about. I'm thinking about your technique now. It seems to be, and we talked about this, an early detection to whether a treatment is being efficacious, if it's actually working. Have you done studies, or has it got to the point yet where you're able to align other ways of looking at that tumor, for example? Like as a tumor shrinks, generally we use MRIs and things like that to monitor it over time. Have you guys got to the point now where you're able to say, okay, we think the rate of decomposition is this, therefore when we next do the MRI in a few weeks, it should be at this point? Is it predictive to other types of techniques yet?
Not yet. So, I mean, it's still really early days. But however, what I will say is that this whole idea of treatment response is really picking up. And there's another competing technology that has just beautiful preliminary data that shows the same type of an idea, and that's something called diffuse optical tomography. So here now you just send light in, right?
Okay.
You detect the light that comes out, how it bounced around, what it did, and because it's very sensitive to hemoglobin in the blood, what
Okay.
the researchers has found is that, again, right, if a tumor responds early in the treatment, those parameters change dramatically, whereas if it doesn't respond, those parameters remain relatively the same, right? So, this entire idea of looking at changes either in cellular structure or in blood supply during a treatment, I think personally is going to be a very powerful paradigm, right, going forward. And if you look at MR and CT, beautiful modalities, good contrast, but they're not sensitive to cell structure or as much blood flow, right? Okay. Whereas these techniques are kind of ultrasound in terms of cell structure and optical techniques in terms of blood absorption are targeting very key areas of what you'd want to know about tumor response.
It's getting more refined as opposed to the brute force of imaging the whole tumor, right?
Exactly, much more refined. You're looking now at the cellular level or surrogates of the response at the cellular level.
Right.
Well, guys, we are, I'm afraid, just about out of time. Dr. Kolios, so as the host of the show, I want to make this very clear that I get to pick who gets the last question, see, of the interview, okay?
Okay.
And
Good.
yeah, I see that, Dr. Merritt. And today I picked me. So here's my question to you.
There you go.
For the last question.
Right.
What fires you? What is your passion? What keeps you coming back to that lab every day, day after day, to run whatever it is that you run in there? Message for the kids out there. What is it that's driving you right now?
Love for new knowledge, right? The thirst for generating new knowledge, but with a little twist to see it also applied, right? So, this, saddles between basic research and applied research. So you could think of this in a very abstract theoretical way, scattering of ultrasound from cells. Somebody might say, well, who cares, right? But I always had that endpoint of I want to understand this because this is what I want at the end of the day, right? So the fact that I love science, but I also could see the application having such a huge impact if it works the way we want it to work is something that could, keep me going 24/7 for 100 years.
Thank you very much. We'll continue to follow your research and, with great interest. And thank you on behalf of everybody, Canadians, students internationally. If this works and if it's developed, you're gonna make a huge impact with this research. So thank you very much for doing that, and thanks for being on the show today as well.
Yep, and thank you for allowing me to, talk to young minds, which are the future of Canada, and I encourage them to pursue their crazy ideas. Do it scientifically, do it rigorously, but pursue them.
Dr. Kolios has joined us today from Ryerson University, where he is a cancer researcher. Thanks, everybody, for tuning in to another episode of This Week in Science and Education. We'll catch you all soon. Take care.